Okay, so we left off yesterday or Friday talking about the three binding sites in a ribosome. So here they are one more time. So this is my large subunit of the ribosome and the small subunit. So a ribosome is actually composed of two smaller parts, but they fit together and bind over the messenger RNA. And then we have three binding sites in the ribosome. The A site, which is where the transfer RNA that is bringing in the next amino acid that needs to be added to the chain lodges. You have the P site, which is the site that holds the transfer RNA that has the polypeptide chain attached to it. And then the E site, which is the site where the empty tRNA will go to be ejected out of the ribosome. So ribosome has two major jobs. Number one, it has to decode the messenger RNA. So it holds the messenger RNA in the right position so that it can be decoded by the transfer RNA. And the other job is to form peptide bonds between the amino acids. So part of a ribosome, because remember, a ribosome is a complex of ribosomal RNA and other proteins. One of those proteins is an enzyme called peptidyltransferase. Peptidyltransferase, again, it's transferring the polypeptide, therefore making a new peptide bond. It's actually the enzyme part of the ribosome that helps make that peptide bond between our adjoining amino acids. So the amino acid that's brought in by the tRNA in the A site will be bound to that polypeptide chain that is being held by the tRNA in the P site, and that peptidyltransferase will make the bond between them. And so, right now our current research says that the ribosomes and all the jobs they do are really kind of taken care of by the ribosomal RNA that makes up the majority of the ribosome. So looking at translation in prokaryotes, there is an initiation complex. So just like we have a transcription initiation complex, there's a translation initiation complex because things have to start the process off. So what does that include? Well, it includes the initiator transfer RNA that has methionine on it, the small subunit, and the messenger RNA strand. So when those three things all come together, that creates the process, or that starts the process of initiation. Excuse me. There is a binding sequence on the ribosome, on the messenger RNA that binds the messenger RNA to the ribosome in the right way and makes all of those pieces fit together correctly, and especially the small subunit. Once that initiation tRNA and the small subunit and the messenger RNA strand are all joined together, then the large subunit will be added to it. And when the large subunit clamps down, that initiation or initiator tRNA molecule will actually be in the P site with an available A site ready to start. So here's my translation initiation complex. You have your initiator tRNA with methionine. There's your messenger RNA and your small subunit. We have this initiation factor and this initiation factor, right? Just like we had transcription factors, we have translation factors. And they just start and get things, they start the process. They help aid in that process to get everything joined together. So here's my initiation complex with that first tRNA bound to the codon, and then my small subunit. And then the large subunit clamps down with that tRNA in the P site. So eukaryotic translation, very similar. In eukaryotes, the actual amino acid is methionine. The other one in prokaryotes is just a little different version of methionine. There is a bit more involved with the initiation complex. We don't, in eukaryotes, we utilize that 5' cap so that the small subunit knows where to attach to the messenger RNA as opposed to a ribosome binding site like the prokaryotes have. So remember we said the 5' guanine cap helps with binding to the ribosome, so that's where it does that. So initiation occurs in that way. And then we have elongation. And so that second transfer RNA is going to bind into the A site. Not really worried about the particular elongation factor. Don't worry about that. But once that A site is full, the peptide bond is going to form then between the amino acid in the A site and the polypeptide chain. And then the process just kind of keeps going on. The ribosome will translocate and move down so that what was in the A site is now in the P site. What was in the P site gets moved to the E site and gets ejected out of the ribosome. And so that process will continue where the anticodon on the tRNA will match up with the codon on the messenger RNA. You'll form the peptide bond between the amino acids. The ribosome translocates or slides down the messenger RNA. So here we have our full A site and P site. The bond is going to be created between these two amino acids. There's a peptide bond there, and now the ribosome will translocate and move to this site. So what's in here will actually slide and be placed into this site here, so that my empty tRNA goes to the E site. And so we're going to eventually create the chain of amino acids. Remember we said that the form of that primary structure of a protein is the NCC repeating backbone. So here's my N terminus. The amino acid has that structure. Remember the central carbon, the R group, the hydroxyl group, right? The carboxyl group. And so then your end of the polypeptide chain is called the C terminus. So here's the whole process put together. Number one, we got the tRNA anticodons match with the codons. Then we have the elongation factors bringing in the correct amino acid on the right tRNA. Anticodon-codon pairing is correct. Then we get the peptide bond formation between them, right? So you can see from here, they are now bonded together, and the ribosome will slide down. Oops. The ribosome will slide down. So what was once in the A site is now in the P site, and we have that growing polypeptide chain, and the process just continues. Now, as far as matching, there aren't as many tRNAs as there are codons. So when you look at that codon chart, there's 64 codons. Well, there aren't 64 different tRNAs. And what we figured out is that the third base on the tRNA has kind of more loose pairing rules than typical nucleotide pairing. So wobble pairing is what we call that. And wobble just means, the third position doesn't necessarily have to match exactly right. And that's how we can account for fewer transfer RNAs than there are codons. And so that process is going to keep going. Elongation is going to continue until there is a stop codon encountered by the ribosome. When that happens, when they find that, when they hit that stop codon, then release factors will enter, and they will basically, release that polypeptide from the ribosome, and everything kind of disengages and lets go. So when the ribosome encounters a stop codon, which is here, a release factor binds into the A site. That releases your polypeptide chain, and then everything else breaks apart.